My name is Zoran Pavlovic, and my research is evolutionary conservatism of a mammalian riboswitch, and I work with Dr. Sokup's lab in the biochemistry department. So what you basically have in biochemistry and biology is this central dogma of DNA to RNA to protein. So you have transcription to translation. And that's what a lot of scientific research nowadays deal with, is the coding regions that actually produce proteins. And what we're interested in is the non-coding regions that actually control the expression of the coding regions, and particularly riboswitches. What you usually have is you have a protein that is produced and it can actually regulate its own coding by binding to its own sequence. What riboswitches do, and what's really interesting about riboswitches, is that a metabolite is produced somewhere within the synthesis pathway, and that metabolite actually comes back, binds to the mRNA or the coding sequence, and actually affects and controls the expression of that gene. And so riboswitches have been very well characterized in bacterial cells, not so much in eukaryotic cells. And so what we're interested in is characterizing the first ever mammalian eukaryotic riboswitch in mammalian cells. So bacterial riboswitches, for example, the TPP riboswitch, which has been well characterized to this point, will contain some kind of an aptamer and some kind of an expression platform, and then the actual coding sequence that you want to control. As I said before, a metabolite will be produced in the pathway that will eventually bind back to its expression platform and then either turn off or turn on the expression of that gene. And so as we can see in the TPP pathway, when enough TPP is produced, it'll bind back to its expression platform and its aptamer and actually stop the production of the THIM protein and of TPP. And so our specific riboswitch that we're looking at is known as antizyme. And in our antizyme mRNA pathway, what we see is that antizyme interacts with an enzyme called ornithine decarboxylase. And ornithine decarboxylase is instrumental in the production of various polyamines within our body, specifically turning ornithine into putrescine, which is a committed step in polyamine synthesis, which goes on to produce spermidine and finally goes to produce our metabolite of interest known as spermine. Polyamines are extremely important in the cell because they're involved in a multitude of cell processes such as cell growth, cell proliferation, cell death. So polyamines are extremely important in the cell. Polyamines are also extremely important in therapeutic studies because, as we know, we've known that polyamines can actually stabilize the negative charge on DNA backbones and therefore stabilize DNA. And this is really important in cancerous cells because if you could somehow destabilize cancer cell DNA, you can probably produce a therapeutic or some kind of drug that can cure cancer or at least help fix the disease. So that's why we're really interested in spermine, polyamines, and the antizyme riboswitch. And so what we believe is that this is a proposed structure, mRNA structure of our antizyme. And the way we believe that this process works is that we have our antizyme mRNA and we have a certain frameshift site. The frameshift usually reads UCC UGA. When it reads UCC UGA, spermine will bind to this and no antizyme protein will be produced from the coding region. But when spermine actually binds to the antizyme, it'll cause a +1 frameshift to CCU GAU, and that'll turn on the coding region and then turn on spermine synthesis. So that's kind of a way to upregulate or downregulate spermine synthesis and antizyme coding, antizyme expression. And that's what we're interested in looking at. And so in this lab, we performed 2 experiments. Those experiments were equilibrium dialysis and inline probing. Both experiments were used to characterize the interaction between our metabolite spermine and our RNA, and so that there's an actual specific interaction between our antizyme RNA and our spermine metabolite. So the first experiment I'll talk about is known as equilibrium dialysis. And what you basically have is a small chamber about an inch large with a small tube about an inch large with 2 separate chambers separated by a semi-permeable membrane with a 5,000 Dalton molecular weight cutoff. Large enough to let our spermine metabolite through, but small enough to keep our RNA to one side. And that's really important because we want to keep our RNA to one side to show that our metabolite of interest goes to the other side, interacts, binds with our RNA, and stays there. So, like I said, if we have functional RNA or functional metabolite that'll interact with our RNA, the spermine, which we label with carbon-14, so we can monitor it, will travel to the other side of the chamber, interact with the RNA, bind to the RNA, and stay there. If there's no interaction between a metabolite and RNA, then what we're going to see is some kind of an equilibrium being reached, which you can again monitor through radiation. And so we performed this experiment And what we got was bar graphs such as this one, which talks about the fraction of partitioning of spermine. And what that means is if you have a fraction of partitioning of about 1, that means that all of your spermine travel to the other side, interact with the RNA, bound to the RNA, and stay there. If you have a fraction of about 0, that means that none of the spermine actually moved and interacted with the RNA. And you have a fraction of about 0.5, that means that there's some kind of an equilibrium that was reached. So when we performed this experiment, in this first bar right here, this is a chamber that had the spermine metabolite that was radio-labeled and an RNA on the other side. What we saw was a fraction of partitioning of about 0.85, meaning that a large amount of our spermine moved into the RNA chamber, interacted with the RNA, bound to the RNA, and stayed there, which is exactly what we were hoping to see. Then what we did was actually try to compete our spermine with non-radio-labeled spermine and non-radio-labeled spermine analogs known as spermidine, putrescine, and cadaverine. So we're doing this because we're trying to show the specificity for our riboswitch for our polyamine spermine. Because if your riboswitch doesn't have specificity, then it won't serve as a good therapeutic target because we don't know what it'll interact with or how it'll work. So as we've noticed in our experiments, as I said originally, is that spermine bound to the RNA. But then when we competed it with cold spermine, we saw that some kind of equilibrium was reached, meaning that half of the RNA bound to radio-labeled spermine, half the RNA bound to non-radio-labeled spermine, which is exactly what we expected to see. Then when we added in the spermine analogs that were non-radio-labeled, we hoped to see that the fraction of partitioning would actually go back to what it originally was, which is exactly what we see, and that's very hopeful. That indicates that our spermidine, putrescine, cadaverine do not interact with our enzyme RNA, and that our spermine does interact with our enzyme RNA, which is really great news. And we performed the same experiment with both mouse, human, and oyster RNA. And the reason why we wanted to do that is because we wanted to show some kind of evolutionary conservatism across a series or spectrum of species of eukaryotic cells. And the reason why you want to show that is that you want to prove that riboswitches not only are very specific to their metabolite, but that they also have some kind of evolutionary conservatism, evolutionary development that is very important to eukaryotic cells. In the second experiment we performed, known as inline probing, the protocol basically, we would originally have to extend and transcribe our own RNA, but what we do now is actually PCR to obtain our RNA. Then we radio-label our RNA with phosphorus 32. We quantitate our radio-labeled RNA using a scintillation counter, and we want about 250,000 counts per minute of RNA radiation. We set up inline probing reactions where we had our radio-labeled RNA, we had our spermine analogs, we had our spermine, and we let them all react for about 3 days. Then we stop our reactions, run the reactions on the sequencing gel, and then put the sequencing gel on a screen, on a phosphor imager screen, so we could actually obtain bands, which is done by the radiation going onto the screen. And so what actually happens in the inline probing experiment, and why we call it inline probing, is as we all know, RNA is self-cleaving, it has catalytic activity. And so when the OH group on the sugar of the sugar phosphate backbone of the RNA comes in line with the phosphorus and the oxygen on the sugar phosphate backbone, the RNA will actually cleave itself and we'll see various bands across the board. But if we add a ligand that binds to our RNA, interacts with it, it'll prevent this self-cleavage from happening, and it'll basically prevent and protect the RNA from cleaving itself. And so we're going to have missing bands or a decrease in the color of bands. And so when we performed this experiment with mouse, human, and oyster RNA, what we wanted to see is that as spermine concentration decreased, that the bands in our gel will go from being light colored, meaning that there was protection and there was no self-cleavage, to being more dark colored, meaning that there was no protection and then there was cleavage. And what we noticed in the mouse gel is that you actually have, when spermine concentrations are pretty high, about 1 millimolar, we have very extremely light bands. And as spermine concentration decreased, you saw that the color of these bands actually increased, which is exactly what we wanted to see, because when spermine concentration decreases, cleavage increases. So this is really good results for our lab. And we performed the same experiment on mouse, human, and oyster RNA, again to show some kind of evolutionary conservatism. However, we did have some issues with some of our gels, such as a salt bridge, pertaining to the conditions and the pH that we use in our gels. And sometimes we would get irregular gels with no pattern of interaction or binding between spermine and our RNA. We did think, however, that we had enough data to actually prove our point that spermine is specifically binding to our antizyme riboswitch. However, we received a pretty strong, pretty powerful criticism of our results and of our data. And what one critic said was that, well, of course, spermidine, cadaverine, putrescine are not going to bind to your antizyme RNA. Look how much they differ in structure from spermine. Because you have spermine, which has 4 amine groups, and then you have spermidine, which has 3 amine groups, and then cadaverine and putrescine only having 2 amine groups. And as we know in organic chemistry, when your structure differs so greatly, obviously you're going to have problems with interaction and binding. So basically, these experiments don't really prove that spermine is specific to our antizyme. So what we wanted to do to kind of put this criticism to rest is run the same inline probing experiments with spermine analogs that we named spermine A, or spermine analog A, spermine analog B, and spermine analog C. These analogs differ enough in structure to our spermine polyamine to show that they are different, but they're similar enough in structure to our spermine analog, similar enough to show that there's not such a large difference in structure to indicate that spermine and our riboswitch is really specific to binding to just spermine and not any of the analogs. So we ran these same inline probing gels with our spermine analogs. And the first gel that we ran, we had the same spermine concentration as we had on previous gels. And what we noticed is that as we increased spermine concentration, the bands in our gels went from being dark to being lighter, showing that as we increased spermine concentration, there was more interaction binding with the antizyme RNA, there was more protection, and there was less cleavage, which is exactly what we hoped to see. And when we ran our analogs A, B, and C right next to that, we noticed that the bands remained very dark, which shows that there was no protection and there was self-cleavage. And so we also performed a gradient gel with spermine analog A and spermine analog C. And the reason why we did it with spermine analog A and spermine analog C is because analog A and analog C are the two polyamines that are most similar in structure with our actual spermine polyamine. And so when we ran those gradient gels, we noticed that there was really no pattern of interaction or of binding or of cleavage, which then again shows that analog A and analog C do not specifically interact with our antizyme RNA, and that our spermine metabolite is specific for our riboswitch. All of which, farther proves our point that riboswitches are specific, riboswitches are conserved, and that our antizyme riboswitch specifically binds to our spermine polyamine. And so future studies in this lab basically consist of continuing our X-ray crystallography experiments to farther characterize and actually determine the structure of our antizyme RNA. Because once you actually get the structure and can determine the binding between the polyamine and the riboswitch, you can then move on to doing in vivo studies and actually developing therapeutics to combat diseases and various cancers and whatnot. We're also going to try to run the same gradient gel that we did with analog A and analog C with our analog B, which we have not run yet, which we expect to see the same kind of results, which is spermine analog B will not interact with our RNA, and so we won't see any kind of pattern of binding, interaction, or protection. And then finally, years down the road, you can do in vivo studies of introducing polyamines into someone with the disease in clinical studies and seeing how that may actually affect the disease and try to cure it or try to act as a therapeutic. And so for my research, I'd like to thank Dr. Sokop and all the members of Dr. Sokop Lab. I'd like to thank the Beatty Lab for helping us crystallize our antizyme RNA structure. And I'd like to thank NIH funding for providing the money for me to perform this research. Thank you.